for the last talk of today, or of this session. I'm Akman, and I'm a postdoc with Akman at UC Berkeley. And today I'm going to be talking about climate-associated adaptations in Arabidopsis thaliana. So, as evolutionary biologists, ecologists, geneticists, we've been long interested in how adaptations are shaped by the environmental cues. A very famous case being the study by Klaus and Kekenheisi, where they looked at two ecotypes, namely Californian and Alaskan yarrows, where they show that the plants, the ecotypes the ecotypes do better in environments that are more closer to their local sites in terms of the environment. And another study comes from the Uxtra lab, where they show that lighter color mice do better in the dunes because they can better camouflage themselves. And these studies provide a lot of insights into how adaptations happen, into the genetic and phenotypic underpinnings of adaptations. But there is also another question that arises from these studies. How global are these patterns? In other words, the adaptations that we detect in, say, a couple, or if you're lucky, in several environments, are these applicable to wider ranges, or even to the whole distribution range of an organism? This is a relevant question for agriculture, for breeding purposes, for conservation and management efforts under global climate change, as well as medicine, as we know that treatments that have been tested in certain ethnic groups do not necessarily work on others. So in the Akman lab, there are several folks who are interested in questions like this, one of them being Jack Colicchio. And this is a study we started about less than a year ago, and we are hoping to put it up on bioRxiv very soon. So if you're interested in this talk, please follow us on Twitter, and if you want to get the updates. Okay, so within this talk, I'm going to we'll try to answer two questions. So the first one is, how does the climate shape gene expression and flowering time across evolutionary scales? And the second, by using these patterns, can we predict flowering time? Going with the first question. So first, I want to give you an idea about the data sets that we use, and I want to make clear that none of this data was collected by us. They're all free, which was really nice. But there's an extensive set of data sets available out there. So the first data set we use is the SNP data coming from the 1001 Genomes Consortium. And we are basically looking at about 600 accessions of Arabidopsis thaliana. And we use SNP to get the PCs for all these accessions from the SNP data set, and then use word clustering, which led us to conclude that there are 13 genomic clusters, as you can see here. And these genomic clusters reflect both isolation by distance and isolation by environment. And the first reason we did this was to correct for population structure. And the second reason is, of course, to look at these patterns within these genomic clusters to see if they're general, if we can generalize them. Okay, the second data set is an extensive whole transcriptome data set published in 2016 by Kawakatsu et al. And these are basically rosette plants which were in climate chambers, and they collected tissues just before bolting at 22 degrees Celsius. And of course, you can imagine when you get a transcriptome data set, it's like thousands of genes expression for more than 600 accessions in this case. And we use WGCNA, which is a clustering method to get to co-expression gene networks, or gene expression modules, as I will call them, or GEMs. So at the end, we had 22 of those, and those could be genes that are regulated together. They could be part of the same pathway, or they could be controlled by the same environmental cues. And next to this, we also use individual genes from this data set that showed associations with flowering time or the climate. The third data set that we extracted from databases is the flowering time data on the very same accessions that we used, that were measured at 10C and 16C Celsius, which we can also extract plasticity data from for flowering time. And the fourth set is basically climate data for the source sites of these accessions. And we had 10 climate variables that we looked at related to temperature, precipitation, and seasonality. And by utilizing and using all these data sets, we are basically trying to detect signatures of adaptation. And we are basically trying to find something like this, where there is a trait of interest that is variable and when measured in a common environment, and the climate of the source site climate of these populations show a correlation with these patterns, and gene expression modules, GEMs, also follow a similar pattern. This is a good indication of local adaptation, and we can say that this climate variable acted on this gene expression pattern or shaped it, which was also reflected on these traits or this trait variation. And we are doing this, we are looking at adaptation through 3 different evolutionary scales. So we are first looking at global patterns. The second, for each genomic cluster that I mentioned before, we are looking at the patterns within each of these genomic clusters, and we are testing if we see general patterns for among all these genomic clusters or variable patterns. Okay, so talking about the global patterns, we basically use the mean values for these climate variables within a genomic cluster and the gene expression modules. And we use linear models to test if we can observe significant correlations. And when we look at this, this is an example. There will basically be 13 points corresponding to 13 genomic clusters that we use. The most predominant climate variables that we show or what we see are significant temperature, seasonality, and summer precipitation. And these usually correlate well with RNA processing, GEMs or gene expression modules, carbohydrate metabolism related ones, cell wall related ones, and glucosinolates. Okay, and looking at the consistent patterns, so this is basically what we're looking at. Within each cluster, we look at these patterns, and if they are variable, if they are consistent patterns, the patterns that we see within each cluster should be comparable to each other among all these different genomic clusters. So it would look something like this, where we see a negative correlation between a gene expression module and a climate factor. Okay? In this category of selection, we see summer precipitation, temperature, annual range to be the most predominant forms of climatic associations, which seem to be associated with GEMs that are related to defense, like jasmonic acid wounding and glucosinolates. And the third form is the variable patterns, where we see within each cluster, we either see a positive correlation for one cluster and a negative correlation for another cluster. This is essentially the climate by genotype cluster effect. And for this type of selection, we see that winter temperature, summer precipitation, and mean temperature are the climate variables that show the most significant associations. And we see gene expression modules that are related to photosynthesis, defense, wounding, response to biotic and abiotic stimulus. Okay, so what I told you is basically within this whole gigantic heat map. And here we have the 3 different patterns, the global patterns, the consistent patterns, and the variable patterns. Here we have the gene expression modules, all 22 of them, and we have the climate variables that we used. So I don't want you to take anything from this heat map, but what I want you to see is that there are all sorts of variations, associations that we observe. Okay, so now we are basically looking at single genes. Okay, so I just want you to focus only on this very first block here. So for the individual genes, we looked at which ones show significant associations with flowering time, and FLC and SOC1, which are known regulators of flowering time, showed up as the most significant genes. And since they're also correlated with each other, we just use them as a single module. And what I want you to look at is when you look at these correlations, the most predominant forms of selection happens to be variable among these clusters. Okay, so then we also looked at these associations for flowering time itself, the phenotype that we observed. So here is 16C, here is 10C, and here is plasticity. And again, we see that the patterns of selection are mostly variable, also for flowering time, which is possibly controlled by this FLC SOC1 module. Okay, so there are several stories that arise from this paper, so please go and check the paper when it's out. But I'll just tell you one of them, which is related to trade-offs between flowering time and defense. So again, FT or flowering time, this is not the gene, this is flowering time. Yeah, well, when we look at flowering time, we see that there are variable patterns. And when we also look at this GEM5, gene expression module 5, we see that it also has variable patterns with summer precipitation. And this gene expression module seems to be enriched for genes that are negatively associated with SOC1. Okay, this was one of the genes that was that showed the highest amount of association, or just the most significant association with flowering time. And when we looked closely, we saw this pattern. So we saw that late flowering is equal to basically a high expression jump. So like this is the gene expression for this gene module, and this is days to flowering. And you can see that for the screen, and these are basically the only northern latitude accessions. For example, from Sweden, we see that there is a very strict correlation. So the ones that are expressing these genes more also tend to flower later. But we don't see these patterns in either the central or the southern accessions. So we're thinking maybe there's, for these northern latitudes, there has to be a mismatch created for flowering time and when the pathogen abundance is high in wet summers. This is just like an informed speculation. Okay, coming to our second question. Can we predict flowering time by using these patterns? So this is first the climate model that we use by basically adding the climate factors that show associations with flowering time. And we can, in this one we have 14 terms, and with this we can explain about 50% of the variation in flowering time. And by using raster layers, we can extrapolate this model to look at the European distribution of Arabidopsis. And one of the patterns that arise, so this is for 10C model, this is for 16C model. One thing that arises here is that we have more diversity under 16C conditions. And another thing is, under 10C, the inland Scandinavian ones here flower earlier than when they're at 16C. And we think that this is because of the vernalization requirements, which somehow might have been accomplished by putting the plants at 10C. Okay. So the next nice part is coming from the using genetic and gene expression data to predict flowering time. So first we looked at the effects of genomic cluster in predicting flowering time, and we can explain a 32% variation of flowering time by using only the genomic clusters, the 13 genomic clusters. Then adding known flowering time associated SNPs, we can explain about 48%, as you can hear, but a combined model between the two can explain about 58%. So this is all gene sequence variation. And for gene expression variation, FLC and SOC1, which are the genes that I showed earlier, can explain 36 and 44% of the flowering time variation. And when we look at the variation that is left out of FLC and SOC1, basically the residuals, and just by using a principal component analysis on those genes that are left from there, we can explain another 16 and 4% of variation. But again, when we combine these four things together, we can again explain a 58% variation, which is similar to what we can explain with the gene sequence variation. But what is even more impressive is, when we combine these two information together, we can explain 70% of flowering time variation in Arabidopsis thaliana. And remember, the gene expression data is collected at 22 degrees. It's not 16, it's not 10, right? So thinking about this, maybe these mechanisms are more robust than what we think. And thinking about that, we decided to look for more realistic conditions. They're not in the wild. But we went to this paper where they mimicked Spain and Sweden conditions in climate rooms for, I think, around 50 accessions of Arabidopsis thaliana. And with that, we can explain 37% of flowering time in these chambers for these accessions for Spain and 77% for Sweden. Okay, so with that, I can just say that there is great value in analysis of publicly available big data sets for inferring evolutionary and ecological patterns. And evolutionary scales at which we search for selection matter, and it's quite variable. And summer precipitation might control defense and FD trade-offs only in the northern latitudes. And climate models can explain 50% of variation, while combined gene sequence and gene expression models can explain another 70. And thank you for listening. And I want to thank all the people who made their data